I'd like to introduce the next speaker in this session focusing on the genetics of pulmonary fibrosis, and that is Dr. Jeff Trent. He has had an illustrious career as a geneticist focusing on cancer susceptibility and working with Dr. Francis Collins while at the University of Michigan. More recently, he is the founding president and director of TGEn, Translational Genomics Research Institute. Previously, he was the founding scientific director of the Human Genome Research Institute as part of the NIH, and now he's going to share his approaches to state-of-the-art care in genetic discovery of complex diseases as it applies to pulmonary fibrosis. So I'd like to welcome him to the stage and thank him for his participation.
Well, thank you very much. Incredible following Mr. Williams. That was just extraordinary. I'm also delighted to have had the opportunity to follow Dr. Kiley because he certainly emphasized the points around partnerships, around the common good that the NIH can do for large-scale projects. And what I'm going to focus on is something that he made reference to in many times during his presentation, which is largely to say that a small group of people can make a big difference, and this group in this room can indeed for pulmonary fibrosis. So I'll speak to the state-of-the-art approaches associated with genomic discovery, but I'll do so in the context of a few other diseases, but I will end, I promise you, in terms of IPF. So I'm at a nonprofit biomedical research institute. TGEn stands for Translational Genomics Research Institute. And 15 years ago, when I left the NIH, Translational was not used in every single component for every single program. But we're a nonprofit biomedical research institute that's focused on generating genomic information, on analyzing information that comes out of your genome, and probably more importantly, on treating patients based on genomic information. So if you're going to treat patients with genomic information, it's important, of course, to have had that Human Genome Project completed. And I was fortunate to be part of the last 10 years of that project when I was a professor at the University of Michigan and left with Francis Collins and became the founding scientific director for the National Human Genome Research Institute at the National Institutes of Health. Now, for most of you, when you think of the human genome, there are experts in this room that can annotate it and discuss it, but there's also individuals here I know for the first time in a medical meeting like this that are saying, so what's the genome again? Help them remind me of that. So let me just race through some thoughts around that very quickly. First of all, it's the completion of the genetic information that each of us have. If you think about it as the instruction manual, the blueprint, your personal instruction manual. And of course, solving and getting that first genome done, it was an exciting time. A little over a decade ago, Congress made a concurrent resolution around Genome Month and DNA Day. It was the only time that Google has changed its moniker for a day. And back then, they had stamps, if you remember those things. And there was one on cracking the code and on comparative genomics and on the end of the beginning, the Churchill sort of quote. And that is the reality of where we were a decade ago and where we are today. 3 billion pieces to the DNA alphabet, and the interpretation is the next phase of medicine, probably for the next 100 years. Well, it's a lot of data to sift through. 3 billion letters in each one of your 14 trillion cells that have a nucleus. And for us to be able to sequence and make clinical decisions, we actually have to sequence the genome multiple times because there's an error rate in our sequencing. So we usually do about 100 billion data points per individual. But then when we finish looking at that, and this is a picture of the Statue of Liberty next to, if you didn't cheat and you took an 8.5 by 11 paper and you printed out in 12.3 billion letters, it would be as tall as the Statue of Liberty. And a single change, a single missing comma, a single misspelling on a single page can really make a major difference in diseases and disorders. And that's what we're sifting through, and the task that some of these remarkable physicians and scientists in the room around IPF are looking at every day. Now, again, it'll surprise at least some people in the room. I could probably name a couple that there are no perfect specimens. All of us are born with between 5 and 50 genetic flaws. And it's important, therefore, for us to look at the information that we're born with and really understand that for disease. But the goal is really that by finding molecular targets before treating disease, not only does medicine become more rational, it becomes more personalized. Now, this is really where I depart from I spent my 10 years in the federal government, and what an amazing opportunity the NIH provides for common good projects. But what a remarkable woman Margaret Mead was, and I don't discount what she said in any regard as being the truth, that you should never doubt that a small group of thoughtful, committed citizens can change the world. It's the only thing that ever has. This group can change the world. Well, think about it, though. If you're at, the NIH is 38, or it was when I left, billion of your tax dollars at work. So what's it like to leave the NIH and start an independent, nonprofit biomedical institute with a few hundred people? Well, I think this is a great picture. This is Felix Baumgartner of Red Bull at 130,000 feet above the earth, about to step off and hurtle down. He actually breaks Mach 1 on the way down. But he lands successfully, so that's the positive part of being able to share that. But it's a big step. How can this group, how can IPF, how can the groups here really join together, leverage, of course, what the NIH is doing, but make a difference? So in our case, we put together programs. This is the scientific version of what we did, the science team, but also right away we brought together physicians to perform clinical trials. We do deploy a lot of technologies across a number of disease indications, including, of course, the lung. We've sequenced more than 500,000 different samples with over 23,000 treatment decisions made in regards to sequencing. And this is a picture of some of the instruments that we use. And the one on the far right that was recently introduced means that what took us 10 years, the first genome project, it was, by the way, a government project that was ahead of schedule and under budget. It only took us $3 billion instead of $5 billion. It only took 10 years instead of 13 years. But 10 years for a single genome, and the machine on the right will at least sequence the genome for you in about 10 hours. So it takes a bit longer than that to analyze the information coming out of it, but just a remarkable difference as we move towards the cost going down to $1,000 per genome going forward. It does mean you need a lot of high-performance computing. We've been very blessed to work with Dell and EMC in the development of something that in our group is a purpose-driven high-performance computing environment, about 60 teraflops of operation. That means it does about 60 trillion floating point operations per second. Our group uses about 1 million CPU hours per month, and we use about 2 petabytes of storage that we have to keep floating at all times. We've worked on a lot of the precision medicine trials in cancer with a lot of the different major collaborating academic institutions that you see here. But we also look at a lot of idiopathic diseases, and especially those around childhood, trying to make discoveries into undiagnosed childhood disorders is a major part of the effort at TGEn. But the reason I'm here in the last few minutes of my talk, I'm going to speak to the role that really I was asked to focus on is how these consortia for diseases, you heard about some wonderful examples from Dr. Kiley a moment ago, can be important, and it doesn't matter if it's bipolar disease or if it's others, and I'm going to focus on one particular in the cancer space because I think it's highly analogous to IPF. And that's the Multiple Myeloma Research Foundation and its precision medicine model. And Kathy Giusti, its founder, decided about 15 years ago, and we were privileged to actually help with her over that time, to say that she would put in place a data bank around samples, like you've heard, and around generating data, be able to put together a learning network and be able to help seed clinical trials. And Clifton Leaf of Fortune magazine is the one who put the famous cover on that said, "We're losing the war on cancer." He said that Kathy Giusti and the MMRF have created the most successful patient advocacy model on the planet. So we've sequenced every single patient in her 1,100 patient enrolled study called the COMPASS trial, generating the data information. And what you see in yellow on those bars are the amount of patients that are enrolled and paid for by the federal government. The green shows you the samples that came from the Multiple Myeloma Research Foundation and their funding. Although I want to assure you that all of the data that was generated, even though it was actually funded by pharmaceutical companies' support and foundation support, went into the public domain and is equally available to any of the government projects. And if you actually say how much data was generated, what's the different tools that we used, I won't go into those. It outstrips anything that the federal government has done, whether it's for breast or lung or others. And again, myeloma is a blood cancer. That blood cancers are 2 to 3% of all cancers of humankind, and myeloma is a fraction of that, and yet they've been able to develop something that's really remarkable. Now, for the scientists in the room, this size data is so important for IPF as well. This just shows an example of one gene. It's called P53, and we've known for a long time if you have this gene and a genetic change, then you may be resistant to treatment, so they really have to treat you fairly brutally. But because we had enough data, we were able to figure out, and again, this is largely for the scientists in the room, that you could have a single change and you would have the same outcome, a single change in one of your 2 P53 genes. If you lost both of them, then you had a very fulminant clinical course, died quickly of your disease. And it really takes large amounts of data to be able to make these small inferences, again, something that Dr. Kiley mentioned. But why is this such a great model for you? This is an example that in the past 10 years, they've had 10 drugs approved by the FDA. They've launched 70 clinical trials, and they've tripled the lifespan of patients by 3-fold. A small group doing a major effort going forward. Well, I want to now really encourage this community that groups are working together in a unique way. You know, only in academics could the battles be so vicious because they mean so little. That's whether you like Henry Kissinger or not, there's certainly some truth to that. But I would argue it in a different way, that only in academics should the battles be so fierce because they mean so much. So this is my personal story, like Bernie Williams gave, a bit different. This is my 33-year-old daughter in the middle. She's with her husband and our 3 grandkids. She was diagnosed a year ago with breast cancer, and he didn't play baseball, but he played for the Arizona Cardinals, who lost last night, unfortunately, but so he is that tall. But just bear with me for one moment. This is some words from my daughter, and it speaks to how important precision medicine is. She said, "To be honest, I'm just adjusting back to normal life. I made it to 3-week point where I would have had chemo, but just had a Herceptin injection." That's because she had a molecular change that defined her treatments to receive that specific drug. She said, "I had 0 side effects beside the taste of saline in my mouth, and I just couldn't believe it. No side effects. I sat there receiving it and had to tell my heart to calm down. Chemo's done, but my body didn't want to believe it." You see, after a while, walking through those doors for treatment is brutal. You know it's necessary, but it's walking towards suffering. So walking through those doors to receive a critical drug that does nothing but its job is hard to believe. Well, I realize and understand, not the way the patients in the room and the families do, that whether it's cancer or IPF, the suffering is just extraordinary. And if we can really leverage our efforts together, we can make a difference around this. So this is my own model of what happened in terms of a consortium meeting on Wednesday that included industry, academia, government, and patient advocates. And really one individual, Tom Hervis, is apparently really working to provide significant funding in regards to what could come alongside government and academia and industry. Dr. Blackwell, Kropsky, Garcia, Kaminsky, individuals from the University of Michigan, Chicago, National Jewish Cornell, the industry partners of Genentech and Behringer, and patients like me and others to just sort of frame out, well, what could an IPF precision medicine model, just step 1, look like? And genetic data, imaging data, investigations to the clinic we're fortunate to be able to begin work. And why do you need a consortium? This is a slide, and then we'll hear more about it from Dr. Blackwell and Kropsky's group at Vanderbilt, looking at genetic information from a large number of families in which we're getting involved at TGEn and just helping annotate that information. And the reason you need a family is this is sort of what the genetic information looks like if you start to drill down on it. And these are all of the different functional changes that you might be looking at. And everyone does it slightly different, working together in consortium, Nancy Cox at Vanderbilt and others, Nick Shork at Our Place and others. These really require teams of individuals, and it's an important component of people working together to look at this large-scale biologic data and try to break it down into meaningful information. But here's another radical idea. Can morphology seen in a medical imaging reveal genetic state? And there'll be a meeting, in fact, that Three Lakes is sponsoring in Germany this weekend, in which radiogenomics is the highlight. And genomics, well, I'll say imaging is the highlight. How do we look at images and look at consistent changes? I saw there was a nice poster from a young physician from the University of Michigan last night in this area. But the area that we add to it is genomic information can make a difference. And that upper left-hand side is Mendel's peas. And this is, of course, is what, you remember they had smooth peas, wrinkled peas. This is what drove most of the people in the room out of genetics, if you remember you're this. But the reality is Mendel didn't know what a gene was, but he could tell smooth and not. He said there's an inherited change that goes with those. On the right-hand side, in 1895, is the establishment of the X-rays, which showed that science could also move towards morphology. So the question is, well, could we use an image analysis, look at size, density, texture, edges, gradients, and actually determine what the genetic information, Mendel did it, why can't we do that? Well, we've been working on that in cancer. There are many groups working on this as well, and we were fortunate last year on the right-hand side to have the paper of the year in the Radiologic Society of North America looking at this as a way, again, to say, well, let's just read through the images, collect those, use all the rapid learning systems, AI, machine learning, but see if we can't then really use that going forward. Now, this is my last part in just the last 2 minutes, and it focuses specifically on IPF. You're going to hear a great deal in the next coming years about the fact that while it took us 10 years to do the first genome, we're now doing the individual genome on individual cells. And remember, you have 14 trillion or so in your body. But it's so important for us to understand the individual cell contributions, especially for a disease like IPF. Traditional expression profiling, that means that we just take a piece of lung and we look at it. We lose critical cell information. We need to look at the biomarkers of single cells. And we work now with a group next to us in Phoenix, Arizona, headed by Drs. Bremner and Drs. Walia in terms of lung transplant for IPF, and a remarkable institute program that does extraordinarily well in the outcome as well as a significant number of cases. But alongside that group at the Norton Thoracic Institute, but also Vanderbilt's group, Dr. Kropsky and Blackwell, with a fantastic young investigator that's here in the audience, Nick Banovich, who was recently funded by the Chan Zuckerberg Initiative to generate SOPs, standards around this technology and put them all into the public domain. It's really beginning the efforts to try to profile molecular signatures using single-cell sequencing. And this picture, which I won't go into, is just says that you can actually do that. In fact, there was a wonderful poster from Dr. Kaminsky's group that showed an initial start at this as well for IPF. It's an important component because we can look at not just your conditions of disease, but whether or not treatment makes a difference, and all the way down to the individual gene level. You're going to hear more and more about this, I assure you. So this is my last slide. Let me remind you again, this group is at the tipping point of really something outstanding. Leverage every bit you can from the federal government, but don't wait for anybody to, and especially not the government, to solve any aspect of this disease. The people in this room have the ability to do it, and I thank you very much for your kind attention.